Whether my slide is visible?
Yes, sir, very much visible.
Okay. Thank you so much. First of all, I would like to thank the organizing committee members for giving me an opportunity to discuss my work over here. I feel proud. I feel really proud of it. So today I'm going to speak about the topic. The gut metabolite trimethylamine oxide is a potent protein folding inhibitor of carbonic anhydrase, a new insight for its involvement in dementia. As Siddharth has also explained something about my lab focus, and basically I'm interested about first of all about the uh, uh, identification of defects in protein folding uh, that is majorly present in many of the brain proteins, especially focused on the neurodegeneration. And in addition to that, we also plan to develop small molecule compounds that can inhibit or correct protein misfolding defect and inhibit protein aggregations, etc. But our approach is little different from there to the others because most of the other compounds that come up to the market or in the clinics, those are of synthetic compounds, number one. And number two, it could be of some phytochemicals that's derived from some plant sources, medicinal plant sources. And others could be some peptide, small peptides that can be because so these are the three strategies that we currently, but our lab focus is little different from those because we wanted to make use of the help of metabolites, because as we know that metabolites are small molecules that's being present in the cellular environment or in the body, and it has certain assigned functions by biological functions of its own. And we wanted to make use of any metabolite that could affect a proper protein folding or that could be of an important cause of the proteotoxic conditions, etc. So there are various types of metabolites. One could be a gut link metabolite, another could be a stress protecting metabolites, and some are toxic metabolites that's being accumulated in the blood, and many other metabolites that's been present. So in fact, because of the progresses in the large front about the metabolomic research, uh, large number of metabolites that relates to the human various human pathophysiologists has been to explore if any of these metabolites could be of that time. So uh, that's the background. So in general, that everybody knows that all proteins that's being a nascent polypeptide chain, they have high entropy and therefore they need to get folded to a native state with the acquisition of the non-native structure, because it's important to come to this native state, because to gain a biological function they need to attain certain 3D structures. This 3D, how the 3D structures look like is being dictated by the non-native structure. That's the common belief that most of the researchers in the field has. And also, but however, quite often there are defects in the protein folding processes that results in the development of proteotoxic conditions, which could be at least three major types. One, um, it can undergo enhanced degradation, thereby leading to the functional insufficiency, or it can sometimes end up in forming high order oligomers, which are spherical in nature most of the time. And another most commonly accumulated or deposited toxic aggregate is called the amyloid, that's being in the brain, that we already observe in many of the neurodegenerative disorder, which are a class of highly ordered and made because of the cross beta seeds, so beta seeds and other beta seeds, and which are made in such a way. And these prototoxic conditions either enhance degradation, the formation of spherical oligomer, or amyloid formation, are known to be the major causative agent for neurodegeneration, various metabolic diseases, cancers, and the atherosclerosis, etc. And interestingly, if we have to look, we have to open another chapter of biology is a gut microbiome, that humans has a gut microbiome. And it also, there are several literatures about the involvement of gut microbiome with these neurodegeneration, metabolic cancer, atherosclerosis, etc. It's been well documented that any dysbiotic conditions leads to neurodegeneration, atherosclerosis, and many other things, etc. So our main curiosity at this point of time is whether any such of gut microbiome, whether they are being linked with this day, is it because of the fact that they're affecting these protein folding processes and they're leading to the development of proteotoxicity, and therefore it's been linked to it. So that's the beginning of the curiosity of it. And so for this we need to look up more about the gut microbiome. So as we know that bacteria causes infections, and we fall sick ultimately because of it. At the same time, that we also teach children that there are also, good bacteria available in and around the system. The most good example is the curd, in which we have the good bacteria. And secondly, our human intestine also harbors large amount of uh, these uh, bacteria, fungus, viruses, and many other organisms. And in fact, these organisms, they provide many nutrients and many other important molecules that our human body cannot synthesize. And also collectively, these are called the gut microbiome. And this microbiota is commonly believed to help in the regulation of host immunity and maintenance of metabolic homeostasis, and they even help to protect us from the invasion by pathogens. And they also provide energy, nutrients, and the many other vitamins that the body cannot synthesize of its own. And the way these metabolites communicate with the host is via the production of certain metabolites called gut metabolites. This could be of various classes or types. It might include tryptophan, indole derivatives, and the branched fatty acids, and the short chain fatty acids, bile acids, and vitamins, etc. So we have a complete database called the gut and gene database that in fact gives about the informations about the completely characterized metabolites in the microbes. So as per the gut and gene database, their existence of 207 metabolites is well characterized, and the 332 gut microbes that we present in the database. So among the metabolites, one such well-studied metabolite in the human is trimethylamine N oxide. So this trimethylamine N oxide, it contains three methyl groups and an oxygen group linked with nitrogen, and therefore positive and the negative charges, making it a deuterion species in physiological existence. These TMAO, in fact, in human is majorly generated from choline or carnitine, which are rich in poultry and red meat, etc. And therefore, persons who are strictly vegetarian eaters, they have very low level of TMAO in them, and the meat eaters and non-vegetarian eaters, they have very high level of TMAO in them. So in fact, in the gut, these choline or carnitine is converted to TMA, trimethylamine, which is a gas form. So in the gut, this TMA is being generated, but when this TMA in the liver TMA is oxygenated with the help of an enzyme called flavin monooxygenase 3. With the help of this enzyme, TMAO is being generated. So many of the individual has PC body odors. That's because of the presence of the TMA. Because many persons that has mutations in the FMO3, they cannot, there is inability to convert TMA to TMA, and therefore feces odor existence is there. So once it's being synthesized in the liver, it's being released to the portal circulation and is then ready for urinary excretion. TMA is not only confined to the blood, as I said now, but can also be picked up by certain tissues with the help of an transporter called organic cation transporter 2, OC2 transporters. But those tissues or cells that is absent of OCT2 transporters, they won't be able to pick TMAO and accumulate in them. And eventually, the tissues rich in TMAO that has been reported so far comprises of kidney, liver, pancreas, etc. So TMAO has been well studied for its ability to fall, and it's been considered for its ability to, in terms of protein folding, and it's been considered to be one of the excellent protein folding enhancing agent because of its attribute to stabilize various proteins and induces folding of unstructured and unfolded polypeptides. It can also correct temperature sensitive mutants. And the previously, way back in 2000 to 2002, uh, while I was a PhD student at the time, we were involved in investigating the effect of these TMAO on different proteins like lysozyme, RNase, and alpha leukoalbumin, etc. So for that, we undergo heat-induced denaturation of the protein in the presence and absence of the TMAO. And this is from the absorbance melting studies, and this is from the by using the CD monitoring using CD. So we see that there is always a stabilization of that because not all the curves has been shown, and therefore 1, 2, 3, 4, 1, 2, 3, 4 represents these different concentrations of TMAO. But here we see that in the case of alpha LA also there is increase in the but as the stability of the protein as the midpoint of thermal denaturation has been shifted toward higher TM values in all these cases. So that makes us to conclude the fact that TMAO is an extensive protein stabilizing agent, and even its mechanism has been widely studied, and it mainly comes out from a laboratory of Bolen and their co-workers. So Bolen's lab, what they did is that they make a very important discovery in the sense that they transfer almost all of the 20 amino acids from water to TMAO solutions. And then the transfer-free energy has been measured for all of the amino acids is being listed here, and including the peptide backbone. So they use diketopiperazine as the mimic of the peptide backbone in these cases. And what they showed is that it's the how TMAO is able to fold protein is because of its ability to overwhelming unfavorable interaction that it has with the peptide backbone. So much of free transfer free energy, around 7 or 8 kilocalories per mole, and other side chains of each of the amino acid does not contribute much. And even most of the charged amino acids, they have negative contributions in the sense that they have favorable interactions. But so it's an unfavorable interaction that the TMAO has with the peptide backbone that is making the protein fall to its native state. That's the discovery. So it's being originated from the overwhelming interaction with the peptide backbone, not with others. So in addition to these, in terms of proteostasis in the endoplasmic reticulum, large amount of work has been done about the ability of the TMAO. So there are at least two possibilities for a polypeptide chain. Say, for example, this is a nascent polypeptide chain, and this nascent polypeptide chain may sometimes it's been converted to a holding incompetent misfolded state, or these nascent polypeptide chain could be a very highly unstable protein. So there are at least two phases by which the proteotoxic conditions arises in the endoplasmic reticulum, as most of the protein folding takes place in the endoplasm. So if this happens to be the formation of incompetent misfolded intermediate, there are at least three fates that these misfolded intermediates they can end up in forming high order oligomers, and that can ultimately lead to apoptosing the cells, and also large accumulation of such misfolded species in the endoplasmic reticulum that can lead to unfolded protein response that's ultimately causing apoptosis again. So in the later slides, I will be explaining about how this unfolded protein response may arise in the later slides. And also these species can also undergo endoplasmic reticulum associated degradation, making it unavailable for the its activity to perform. And if it happens to be an unstable protein, but it can also undergo similar to these, like proteasomal mediated degradation enhance, and then it can also cause the unfolded protein response, ultimately development of apoptosis. But these red triangles representing TMAO, that if we throw in TMAO, that it will be able to shift these incompetent misfolded spaces and transfer to a holding competent intermediate, thereby allowing to fall in its proper native fall, and then vesiculation from the ER, and then transport to the Golgi for the protein transportation. Similarly, for the unstable proteins also, TMAO can sufficiently stabilize it, and making and gaining the ability to form such holding incompetent Golgi apparatus. So that's how the development in terms of the TMAO's ability suffer about the cellular aspect as well as about the in vitro studies. Uh, TMAO, a high level of TMAO in human has been found to be associated with various diseases, including neurodegeneration, various type of cancers, not only colorectal cancer, there are very type 4 or 5 types of cancers, then chronic kidney disease, metabolic diseases, etc. There are not many. So, but recently, its association in the dementia has been alarmingly increasing. That it's one of the causes of dementia. But however, there is no knowledge about whether it's only a bystander molecule, or it's being, one of the causative agent of the dementia. It's not being yet explored. So for that, we come back to the literature, and we found that there are at least 4 different major proteins that's found to be responsible for the dementia. One is amyloid beta, that everybody heard about, that it causes Alzheimer's. Alzheimer's is also a part of the dementia. It's deposition of these aggregates leads to the development of the dementia. And acetylcholinesterase, it's an important enzyme that's responsible for the cleavage of the acetylcholine available in the synaptic, and therefore is responsible for the synaptic transmission. And tau, abnormal hyperphosphorylation, is considered to be the major issue leading to the dementia and the development of neurofibrillary tangles because of its aggregation. And importantly, there is one enzyme called carbonic anhydrase that plays an important role. So we thought that this carbonic anhydrase, in fact, it's been shown to have a neuroprotective role when it is elevated at high levels are available. When its level goes down, that's considered to be dementia. Though all these proteins are contributing to this, so we thought to start with the carbonic anhydrase. There are several reasons why we wanted to start with carbonic anhydrase. Let me come back one by one. So one of the reasons that I said its low level is responsible for the development of dementia is because of the experimental insight that's been shown by previously, that they took acetazolamide and they injected in mice, and which is in fact carbonic anhydrase inhibitor. So when they injected the mice with acetazolamide, and they find that there is impairment in the spatial memory of the animals. Alternatively, when they throw in the carbonic anhydrase activator, that is diphenylalanine, and they observe large improvement in the learning and the memory process, etc. So that's the beginning of the carbonic anhydrase history. Uh, there are in fact 15 isoforms of carbonic anhydrase in human, and in CNS we have most predominantly upregulated form is CA2, CA5, C7, 4, and the 14 are also abundantly available in the CNS. So, but most abundant form was CA2, and therefore it's so we plan to study the effect on the CA2 only. This is how it could relate to the neuronal function about the carbonic anhydrase. Let me just give a glance, because the function of the carbonic anhydrase is, it's a hydration of carbon dioxide leading to the formation of protein and bicarbonate in the system, and therefore it's being responsible for the regulation of extracellular pH. I hope my voice is audible. Hello, anyone, please? Am I audible to everyone?
Yes, sir. Yes, sir. Very much audible, sir.
Yes, sir. Very much audible.
Okay, great. Thank you. I just wanted to test that in the middle, that people are hearing to me or not. So once this bicarbonate has been formed in the intracellular environment of the neuronal cells, the formation of these bicarbonates, the bicarbonate are being thrown out with the help of a transporter. This is a co-transporter. So once bicarbonate is thrown out, then sodium gets in. And also this bicarbonate is thrown out and the chloride gets in. So, and therefore, cellular environment, there is accumulation of sodium ion and the chloride ion, which is responsible for the development of the action potential in the cells. So if we inhibit carbonic anhydrase, there will be no bicarbonate formation, and therefore there will be no development of action potential. And that's the reason why the development of the dementia exists. So it's this carbonic anhydrase, not only from the neuronal point of view, but also from the protein chemistry point of view, that many of the real-time protein folders, these carbonic anhydrase has a focus because of the fact that it is a kind of slow folding protein. So there are two types of protein folding, in fact. One could be in the thermodynamic control, another could be in the kinetic control. In the case of thermodynamic control, protein folds in a wink of eye. That means in terms of microseconds, that we don't know that it's been folded. But however, in the case of kinetic control, it takes a very long time, in the sense that there are reports that, say some of the proteins that take exceptionally completely 3 days to come back to the 9. I don't mean to say that such proteins exceptionally fall in this way, but many of the protein requires longer time, and therefore they are prone to get aggregated. And is the reason why many of the real-time protein folders, they wanted to know about the folding behaviors of the slow folding proteins. And let me come back. One important attribute of the slow folding protein is that they contain large number of proline residues. For example, our carbonic anhydrase contains 17 proline residues. Those proteins that I have shown in my previous slide, that lysozyme, alpha-lactalbumin, and ribonuclease A, those are the proteins which are of fast folding type, because they contain only 2 to 3 proline residues only. So it's important for us to know that why these slow folding proteins, they tend to have more number of proline residues. Let me come back now. So for that, we will need to go back to Ramachandran's discovery now, because it was Ramachandran that shows that every amino acid can have at least 2 conformations. Out of the 20 amino acids, almost all of the 19 amino acids, except proline, they necessarily exist in the trans form because of the fact that the cis form, they experience steric hindrances. But however, for the proline, it is vice versa is correct, because either the cis forms also, they experience steric hindrance, and the trans form also, they experience steric hindrance. And in both cases, either the cis form and the trans, the energetic barrier is very, very low, and therefore it keeps vacillating from cis and then to cis to trans. And therefore, for every native chain, native protein, say for example, a particular cis trans configuration that it exactly needs, that so that the larger the number of the proline residues in them, probability becomes lesser, and therefore is the reason why it is folding slowly. Say, let me come back with an example. Say we have a polypeptide chain, we have 2 proline residues, and this is the native state, folded form of the native state, and we want this side should be a cis form and this side should be a trans form, that it should look like in the active form of the native enzyme. But when it gets folded, there are several possibilities. This P this proline is cis, this is cis, this is trans, this is cis, and this is trans, this is trans. And lastly, cis and the trans, the way that the native state should look like. So this is the actively folded native form, but these are all wrong conformers. So the polypeptide has to keep on vacillating all these things. So if you increase the proline, then there will be many more possibilities, and is the result it will be folding like. And once any of these incorrect prolyl isomers has been stabilized in any native form, then energetic barrier becomes too large, around 20 kilocalories per mole. And here, that's the reason why it requires the involvement of peptidyl prolyl isomerases. I think many of you might have heard about. They are also a class of chaperone called peptidyl prolyl isomerases. They catalyzes the correct conformations of the proline in the peptide chains. So without the help of the peptidyl prolyl isomerases, anything in that ring stabilized in the cis form cannot be converted to the trans or vice versa. And therefore, there is a slow reaction of the process. So with all these reasons, we thought that since TMAO has been one of the important chemical chaperone, as I explained above, we thought that we should try to investigate the effect of these metabolite on the carbonic anhydrase. So first of all, we undergo urea-induced denaturation study because of the fact that, because as I know, as everybody knew, that protein exists in the equilibrium between native and the denatured state. And the equilibrium, that is delta ZD naught, can be exactly understand by knowing the delta Z, and the stability of that can be understood by knowing the delta Z of the proteins. For that, we undergo urea-induced denaturation studies of carbonic anhydrase in the absence and the presence of different concentrations of TMAO. We analyzed the curves for melting concentration and the delta Z values, and we see that there is not much difference in the delta ZD as well in the CM, that it's been reflected that there is not much differences in that, because this is the native state and these are this is in the absence and these are in the presence of TMAO there. So in the reason that it says that it does not change the native state stability, or the equilibrium does not shift because of the addition of the TMAO. If this is the case, it should be reflected in the activity. So we measured percentage activity. As expected, there is not significance in around 15% decrease in activity, but which are not significant enough. So these results led us to conclude that TMAO does not influence the native state structure and the stability of the proteins. But however, when we undergo refolding studies, this is a folding study, these are effect on the native state. Now, in this case, we undergo refolding in the sense that we denature the protein first and allow it to fold in the presence of different concentrations of TMAO. And here we see that this is the refolding control. However, in the presence of different concentrations, there is decrease in the activity, and it's been completely abolished when there is 500 micromolar of the TMAO in the system. So these results indicates that TMAO is a specific folding inhibitor of carbonic anhydrase. So if this is a specific inhibitor of carbonic anhydrase in the folding and it is not affecting the native state, then it should not affect the structure of the native state structure in the presence of TMAO. For that, we use several spectroscopic probes, including far UVCD, near UV, and the tryptophan fluorescent, ANS fluorescent. So these are far UVCD that speaks about the secondary structural content of the protein, that we see that there is not much changes in the secondary structures of the protein. And also the near UVCD speaks about the gross tertiary structural level, which is also not seen to be entirely different because the spectrums have identical behaviors. And the tryptophan fluorescent that speaks about the exposition of the different chromophoric groups. So the chromophoric groups are not at all affected, their microenvironments remain intact. And as a result of it, that ANS binding, because ANS is specifically a hydrophobic dye that we specifically use it when some conformational changes occurs, and if there is exposition of hydrophobic groups that specifically bind to the hydrophobic group, and that can detect that there is existence of some hydrophobic group. So absence of ANS binding and also the no senses the activity is the fact that there is not much difference in the binding of the ANS in the protein. So we also go for, if this is the case, then it should not bind, TMO should not bind to the uh, carbonic anhydrase. For that, we again undergo isothermal titration calorimetry. Here we see that in the when there is excess of ligands up to a ratio of 1.2 molar ratio, there is no equilibration has been popped up, and the amount of energy it's not sufficient enough. That indicates that it's not binding to the uh, native protein. We also undergo site-specific docking at the catalytic site of the carbonic anhydrase, and we observe no binding of the Ca in the carbonic anhydrase. And then in order to investigate whether this activity is confined to TMO only or any other gut metabolites, since TMO is a gut metabolite, so any other gut metabolite also uh, behaves in this way. So we have chosen one or two molecule for each of the classes of this, say, choline metabolism, and then short-chain fatty acid, tryptophan metabolism. So all classes we took 2 to 1 example each, and we found that this is the effect representing the effect of the native state, and we found that there is partial increase or no effect at all in many of the system. But however, there is no inhibitory role of any of these metabolites. Secondly, when we undergo refolding, because I said that TMAO basically inhibits the folding process, not the native state. So in the native state, similar behavior is seen with all the, but however, then we undergo for the refolding process. But we found almost all of them are increasing the folding behavior of that. So these result clearly indicates that the inhibitory role of TMAO, inhibitory role of the gut metabolite is confined to TMAO only, as no other metabolites are inhibiting the carbonic anhydrase. So, as I said, we were also interested to investigate the mechanism how it is being inhibited by the process. So, the first curiosity was that as I said, it's an unfavorable interaction that I talked about. This peptide backbone has an unfavorable interaction that's specifically called the osmophobic force. So, we wanted to verify whether osmophobic force is, if osmophobic force is working, then it should stabilize the protein or it should induce folding of the protein. So it should be the other way around, that the peptide backbone should not be predominating in the system. So using the free energy described for each of the side chains of the amino acids, we were basically focused on charged amino acids, because charged amino acids basically contribute to the favorable interaction. So we count the number of charged amino acids in the carbonic anhydrase and the peptide backbone. We see that positive charged amino acids, it contributes positive, and the negative zone and the negatively charged amino acids, their contributions, favorable interactions are. But however, the peptide backbone contributions is large, outweighing the contributions of this. So it means to say that it should stabilize, but rather here we see a destabilization or a inactivation of the enzyme activity. Therefore, this osmophobic force is not accountable for the destabilizing effect of TMAO or the folding inhibition by TMAO. It's a completely different kind of thing. So we further wanted to investigate for it. There are several possibilities for the loss of activity, that it could be that there is loss of structural integrity, there is oligomorphism, but loss of structural integrity, as I talked about, there is no loss of structural integrity, as I said, I've shown in the previous slide. There could be that in the presence of TMAO, there is oligomer formation, or they can be kinetically trapped somewhere, and there is disruption of the folding intermediate, something like that. So we plan to investigate one. So for the second possibility, that it is forming, because as I explained in the first slide, that many of these intermediates, they tend to form higher order oligomers that results in the functional insufficiency. So we'll look at the light scattering as well as transmission electron micrograph, because light scattering is one of the sensitive tool to analyze the protein aggregation, because it basically monitors about the increased size, because the larger the size of the oligomer, the larger will be the light scattering intensity. But however, we see that there is, in the absence of TMAO, this much of light scattering is there. However, in the presence of TMAO, there is no significant difference in the light scattering intensity. That means to say that the samples in which we observe inhibition of activity by TMAO, it does not show any oligomer formation. And this being confirmed in the electron microscopy, that there are no presence of any aggregate species present in the samples. And then we looked at whether they are kinetically trapped somewhere. For that, we analyzed the activity at different time intervals: 16 hours, 48 hours, 72 hours, and the 96 hours. And we see barely any activity is present at all the time intervals. And also, the loss in activity might be that it may end up by forming aggregates. So for that, we again go for light scattering analysis at all these time frames. And this is the electron micrograph representing at this 96 hours intervals only. So these data completely revealed us the fact that it's not kinetically dependent, because if it is kinetically dependent, the activity is expected to increase after longer incubation up to 96 hours. It might be sufficient enough. So we have not gone more than 96, because after 96 hours, there is appearance of precipitates, and therefore we have not extended. The folding of the carbonic anhydrase has been well understood and to many of the biophysicists, and it consists of two steps: a slow step and a fast step. There are three different species involved. One is the native conformation, and the last is the completely denatured form, that is the US state. This US state is characterized by the presence of incorrect prolyl isomers, and is the reason why they take time to come back to the UF. And the UF, on the other hand, consists of these prolyl isomers that is similar to that of the native state. That means all prolyl are in the correct conformations, and therefore once the UF is formed from the US state, the enzyme can all of the UF molecule can easily be converted to the native state, because creation of the proper or correct prolyl isomers was the rate limiting step of the folding of the carbonic anhydrase. So once UF is formed, then the native state will be generated. But here we could not find generation of the native state. So we thought that TMAO might have targeted this UF. Most probably it might have disrupted and converting to some US species. So this is from the kinetic studies, and in thermodynamics, we can also, in equilibrium, we can also populate, we can also create US and the UF if we allow long-term denaturation in the presence of 5 molar ZDMCl more than 1 hour, that results in the formation of US. So all other things that we have been treating with the ZDMCl and the denaturation studies, those are with the US state only, that we created US state and allow the protein to fall. However, if we change it, we pulse it only with the only for few seconds, few 10 seconds at 2 degrees C, that results in the development of the UF state. So since our curiosity was to investigate if UF is disrupted by TMAO, so we generated UF state in this way by using this protocol, and added TMAO in it, and then we examine the activity of that. Here we see that in the absence of TMAO, protein was folding very well. However, the UF state was folding very well to its native state. However, in presence of TMAO, there is no more increase in the activity. That means to say that the UF state could not be converted to the native state, rather it's being converted to the US state. So this clearly confirms that yes, the effect of TMAO is via affecting the integrity of the UF state of the protein. So in order to confirm what's the exact structure of the state being generated by TMAO, so we use optical probes again. This is the far UVCD secondary structure. This is the far UVCD tertiary interactions, and the tryptophan microenvironment and the ANS binding. Here we see that this is the refolding control that we have, and this is the US state, that's the completely unfolded state of the protein, and however it is in the presence of TMAO. So there is some structure, but it's close to the US state, but not close to the refolding control. But it's highly disordered. But however, there is some differences in the secondary structures. This has been confirmed here also. It's more of the structure has been completely melted, and it looks like to be similar to that of the near to the US state of the protein. And but however, there are presence of certain structures in this case. This is being depicted here also because there is certain blue shift. There is certain blue shift in the case of TMAO treated one as compared to the US state, indicating that there is burial of some of the chromophoric group to the non-polar environment, and this is more of in the polar environment. In this case, there is more of these differences. There are not much differences in the interactions between the so ANS binding affinity. That means the amount of hydrophobic group exposed are not entirely different from each other. So we basically call this state as the UTMAO because of the fact that this is little different from that of the US state of the cell, US state. And then because we thought since it is more towards the US side, that consists of more of incorrect prolyl isomers. So we thought that our TMAO induced structure could contain more of this incorrect prolyl isomers, and therefore that cannot be converted to the protein to assess folding with the help of PPI, as I described earlier. This is one of the cyclophilin, that is one of the peptidyl prolyl isomerase that can correct the inactive prolyl isomers. Surprisingly, we see that is the refolding control. We treated the TMAO in this sample with cyclophilin, and we found it with increase in the concentration of 1, 3, and 6 micromolar and 11 micromolar. There is large increase in the more in the refolding control. So there is large increase in the activity. So this clearly indicates that TMAO was impeding the activity by affecting the cis-trans isomerization step of the carbonic anhydrase. And but however, we see that this is at 11 micromolar. We extended for other time period, but there is no more increment because this has been carried out in 16 hours. We extended 44 hours and 72 hours, but there is no more increase in the activity. We perform another alternative experiment. If this is really true, then cyclophilin can be inhibited by using inhibitor of cyclosporin. That if this is really true, then addition of cyclosporin must be able to impede the process. So in order to do that, we perform this is cyclophilin and the cyclosporin control. It's not giving any activity. But when cyclophilin is added, there is increase in activity. Cyclosporin is not. And when cyclosporin, cyclophilin are added together, this cyclophilin is inhibited, and eventually there is no more activity again. So these results clearly indicates that TMAO affects the cis-trans isomerization state of the carbonic anhydrase, and therefore impede this folding process. So again, our next curiosity is this effect of TMAO confined to carbonic anhydrase only? So for that, since one of the property of the slow folding protein, as I stated above, is that the slow folders, they tend to have more number of proline residues in them. So we have picked up 2 additional proteins, host radius peroxidase and the alpha chymotrypsin, which consists of 11 and the 9 proline residues. And we investigated the effect that here it is that in the case of alpha chymotrypsin, we see that similar to TMAO, there is not much significant effect on the melting temperature or stability of the proteins. Ultimately, native state activity is not impaired. And also the native structural integrity is also not perturbed at all. So this is exactly similar way that is. However, upon refolding, there is decrease in the activity. So this again proves that in case of alpha chymotrypsin also, it is inhibiting the folding process, not the active state stability. But however, in the case of proline, in the case of horseradish peroxidase, there is certain dissimilarity, though refolding does decrease the or inhibit the activity, but native state is also affected, because when we undergo heat-induced denaturation study, we cannot completely measure the complete transition curve, because since it's a very stable protein. But here we see that the nature of the transition curve, that it is going from high TM values to low TM values, that it is destabilizing, and also it is affecting the native state activity. But though there are no significant effect on the structures of the protein. But however, ultimately there is a decrease in the inhibition of the activity upon refolding process, more than that of the native state. And therefore, this again proves that this horseradish peroxidase, though it little in the dissimilar way, it also impedes the refolding process. So there are also existence of various other enzymes that has been reported in literature to be, activity inhibitor or a denaturing or a folding inhibitor. So many data are there about the effect of TMA on various proteins. But when we looked at, but almost all of these proteins tend to have large number of proline residues in them, except one pyruvate kinase that it says that no enzyme activity, there is no change in enzyme activity that's been reported. So all these results led us to believe that TMAO could be a specific inhibitor of proline-rich proteins, or specifically inhibitor of the slow folding proteins in general. So if we have to translate these results in the cellular system, what it could lead to is that it may lead to the evocation of the unfolded protein response, which might lead to the apoptosis that I talked earlier, because of the fact that most of the protein folding, as I talked earlier, that it takes place in the endoplasmic reticulum ER. In the endoplasmic reticulum, there are 3 receptors: ATF6, PERK, and the IRE1. And the GRP98 is a SSP70 chaperone, and they bind to all these 3 molecules, ATF6, PERK, and the IRE1, and inhibit their functions. So their activity remains inactive upon binding to GRP70. Well, since it's a SSP70, they also have a tendency to bind to the misfolded proteins. And therefore, there is a competition that if any large increase in the misfolded protein generation in the endoplasmic reticulum, that in that case, they are being released from the receptors and try to bind to the misfolded spaces. In those cases, once they are being released from there, they started processing each of the receptors, and thereby generating their respective transcription factor, ATF6 for this one and the ATF for this one, and for XBPS, that get into the nucleus and started transcribing genes responsible for the extent of the that might eventually lead to whether it has to go to the restoration pathway or it has to go to the apoptotic pathway, depending on the situation. So when there is an increase in the misfolded species, then there will be a generation of the unfolded protein response. And this unfolded protein response is the leading factor towards the initiation of the apoptosis by the cells, if uncontrolled. So since TMAO is inhibiting these proline-rich proteins, it might result in the accumulation of large amount of cis proteins in the endoplasmic reticulum that might result in the unfolded protein response. For that to check up, first of all, we took HeLa cells and treated them with different concentration of TMAO, 100 micromolar, 250, and 500 micromolar. And so we do 2 key experiments. One, this has been this TMAO has been put in the media while cells are being grown, and in this one we collected the protein samples and then added TMAO in it just to verify it. And then we checked that if there is an increase in the level of the unfolded proteins. So the increase in the level of the unfolded protein was being monitored by ANS binding, because ANS is a dye that specifically binds to unfolded protein. So this is the control ANS, and upon TMAO, this is the control cells that it was not showing anything. However, when the background had been adjusted with the control cells, and with this there is increase in the, upon treatment, there is increase in the ANS intensity, also with increasing concentrations of that. And similarly, with the cells whose protein has been extracted, and after that TMAO has been added, there is also more increase in the binding. So these exclusively proves that TMAO helps to unfold many of the proteins, and therefore ANS intensity goes. So if this is the case, there should be, if there is more increase in that, there should be cell cycle arrest. So in order to check the cell cycle arrest, we go for analysis of the cell cycle phase distribution in the absence and presence of TMAO at two different concentrations. And this is at G0 to G1, 74, and the S is 11, and the G2 by M is 13.4. And we see that at 200 micromolar, there is not much similarity between each of the populations of G0, S, and G2. However, when we added 500 micromolar TMAO in that, there is large increase from 11 to 20, and an eventual decrease in the G2 by M phases. So this clearly indicates that the cells has been halted or arrested at the synthetic phase, S phase only. So this data completely again reveals the fact that the inhibition, so it might also, TMAO could also cause an unfolded protein response, leading to the cessation of the cell cycle arrest. So with these, our major conclusions are that TMAO alters and inhibits its folding. And the results also indicate that since carbonic anhydrase is one of the important protein responsible for the dementia, this could be involved with the dementia. And since large number of data has taken over that there is close relation between increase in TMAO level and the dementia. So the major link is via its inhibitory role on carbonic anhydrase. Though we need to work on the other proteins that that's being introduced earlier. The inhibitory effect on carbonic anhydrase appears to confine to TMAO only as a part of the gut metabolite, because other gut metabolite, as we tested, does not show any significant inhibitory effect on either in the unfolding process or in the native state. And the TMAO could also be involved in the unfolded protein response signaling and also induce cell cycle arrest. So this is my laboratory at present in SCBR, and this particular, all this work of the protein folding and the ball biophysical work has been performed by Ms. Kritika Kumari, and this guy Aniket Kumar Pansal, he basically took the responsibility of the cellular businesses that are being calculated. So with this, I end up my talk. Any questions, please? Yes, please.
Thank you so much, sir. Very nice and informative talk, very fruitful talk.
Thank you so much for inviting me also.
And now the time of question answer. If anyone want to ask any question, then please raise your hand or text in the chat box to write your question.
I can speak also, not an issue. Anybody can use either the chat box or you can just raise your voice. I think there are no more concerns.
Yes, sir.
Yeah, so dear participants, if you have any query, please inquire it or raise your hands, or directly you can unmute yourself and ask the question, so that it will be a great pleasure for us to listen your queries. Striking contrast over the presentation delivered by Professor Singh sir. It was very informative, outstanding, and enjoyable lecture. It is a boon for our young scholars to follow the methodologies and techniques, most recent and advanced sophisticated techniques utilized in the research field to make it more fruitful and societal applicable. And the things that were presented by Professor Sir is outstanding. So I wish the students, especially the young scholars, joined this session as get benefited. So please ask your striking contrast to make it more interactive.
I think there are no more questions.
Yeah, okay.
Yes.
Thank you so much, sir, for giving your valuable time, even though you have very much busy schedule and administrative duties over there. But you accepted our proposal and invitation and given your valuable understanding and enjoyable lecture. Definitely, it was very beneficial for us. We have learned a lot by these techniques. It is quite more advanced and numerical techniques that you have been used here for making it more applicable towards the society and mankind. So thank you so much, sir, for your valuable time to
Thank
share
you
with
from my
us.
side also for giving me an opportunity to talk here. And also thank you for inviting me in this platform again. Thank you once again, Siddharth. We'll keep meeting. All right.
Thank you, sir.
I think I can log out now. Okay.
Sir, I hope now proceed program at next as our schedule.
Yeah, definitely, Siddharth. As per your schedule, you have to proceed for a next proceeding.
Sir, we have already our next guest speaker as a keynote speaker, Dr. Deepti Sharma ma'am already joined. So now I am warmly welcome you, the Dr. Deepti Sharma ma'am from the coldest city, -4 degree temperature at Durham, United Kingdom at 6:18 a.m. Dr. Deepti Sharma ma'am recently joined postdoctoral research associate at Durham University, England, United Kingdom. Dr. Deepti Sharma, a PhD researcher at the Department of Chemical, Biological, Radiological and Nuclear Defense, Institute of Nuclear Medicine and Allied Science in Delhi, is a meteorologist prospects who excel at coordinating many priorities while maintaining and optimizing outlet. Her work, experimental efficacy and safety study of ethyl pyruvate in radiation burn and excision wound model, earned her the degree of PhD from INMAS DRDO, Delhi. She was complete master at the Department of Bioscience, Jamia Millia Islamia. Her area of expertise including cell culture, molecular biology, biochemistry, and wound healing. She has finished publication on the following topics successfully: healing efficacy and dermal toxicity of topical silver nanoparticles loaded hydrogel in Sprague Dolly rat models, DNA binding and anti-radical potential of ethyl pyruvate. In 2020, she filed 2 patents with her name, a composition exhibiting radioprotective and radio mitigating property and process for preparing the wound healing composition are the subject of her patent submitted and Indian patent with applications. She volunteering continuing education program on new frontier in nanomaterial research held in 9 to 12 March 2021 at Department of CBR and Defense. She is a life membership of Vigyan Bharati, member of American Chemical Society and member of Wound Healing Society Cell. Dr. Deepti Sharma ma'am is a very enthusiastic and multi-talented scientist and well-trained researcher in multiple areas such as drug discovery approach using high throughput in silico software, in vitro and in vivo animal model using skin infection injury as a model system. For the past 7 years, he has been working in biomaterial in the field of trauma and casualty care, including wound care and medical management of CVRN emergencies. Dr. Sharma is acquainted with numerous biological techniques and published her research paper in many reputed peer-reviewed journals. She has proved her extraordinary communication skill by presenting her work at various international and national conferences. Now I would like to invite our plenary lectures speaker, Dr. Deepti Sharma, ma'am. Please, ma'am.
Thank you, Siddharth.
Thank you so much, ma'am.
It's a very long speech.
Ma'am, it's you deserve.
Thank
Actually,
you.
your work.
Yeah, it's very cold here actually. And should I present?
Yes, ma'am, please.
Let me know. Can you see my screen?
No, ma'am, actually not visible.
Not visible now. And now?
Yes, ma'am, very much visible, very much.
Okay. So, actually, I am a postdoctoral research associate at Department of Biosciences in Durham University. And I am not allowed to present their work. So I am presenting my PhD, some of my PhD work here on topic prophylactic action of topical hydrogel consisting of anti-inflammatory agent against radiation dermatitis in Sprague-Dawley rats. So as we know, there are various radiation exposure and contamination scenarios, which includes accidental nuclear events, dirty nuclear scenarios, nuclear bombing, radiotherapy, and occupational exposure like mining. And it consists of two types, like local exposure, which occurs through skin, and whole body exposure. It also occurs through skin, but it can be ingested or extravasate the wound, and through inhalation, it can also contaminate the whole body. And there are two types of effects of radiation on macromolecules like DNA and proteins. Whenever a person get radiated or got radiation from various sources, it can the effect can be direct or indirect. In direct effect, there can be single strand break or double strand break. Uh, and in indirect effect, there is a radiolysis of water, which causes the generation of free radicals, which in turn causes the disruption of DNA double helix. And in proteins, the protein can be fragmented, or it can be from protein adducts like carbonylation formation and disulfide bond linkages. And both the effects are very deleterious or harmful for the health. And now proceeding towards the radiation dermatitis. In radiation dermatitis, there are of three types, like acute, chronic, and some have delayed effects. Acute means there is a long time exposure of radiation, and it's produced the effects in within 5 to 10 days. And the chronic effects, when you got the radiation for longer periods of time, and then the effects come to the skin or any other organ. Delayed effects includes when you have got treated for the radiation dermatitis, but after some times it goes it shows the effects after sometimes like 3 months, 6 months, like it happens in the Chernobyl incident. The people gets the keloid formation after 6 months of getting radiation exposure. What is the difference between normal wound healing and radiation wound healing? In normal wound healing, there are 4 processes like inflammation, proliferation, remodeling, and then we get the mature healed wounds. And there is a time period in which it can be performed. While in radiation-induced fibrosis, the ionizing radiation damages the skin through acute radiation injury, and it can take months to proliferate or heal. and after progressive years or progressive time lapse, it can be cause of progressive fibrosis, in which there is a scar formation and continuous inflammation. Sorry, continuous inflammation cause this skin that does not heal on its own. So therefore, it requires some intervention here, like some anti-inflammatory agents, so that the skin can heal properly. So radiation dermatitis have some physical and pathological changes. It divided into four grades, like grade 1, in which there is only erythema. In grade 2, there is a erythema with the dry desquamation. In grade 3, there is also an initiation of moist desquamation. And in grade 4, it consists of ulcers and necrosis. So these are the some interleukins or cytokines which are involved in the inflammatory action during the radiation dermatitis. Whenever cell gets the radiation, ROS generated, either it goes through apoptosis, which is engulfed by macrophages and which generate the cytokines like TGF beta and IL-10, interleukin 10. And either it goes to necrosis or necroptosis, which is engulfed by lymphocytes, which generates the NF-kappa B molecules, and which generates these particular cytokines. And it activates the COX-2 pathway of inflammation, which causes the formation of ROS and then thereby DNA damage. And more of the processes progressive through the steps. These are some wound healing medicines or dressings for radiation dermatitis which are under clinical trials, like Aquaphor, which is effective in treating in prophylactic as well as therapeutic effect. And there is Cavilon. It decreases the inflammation. It only has prophylactic effect. Calendula ointment. It has prophylactic effect. Aloe vera gel. It has to be shown to be a therapeutic effect, but no prophylactic effect. Beta-methoxam 17 valerate cream. It is efficient for prevention of radiation dermatitis, so it has prophylactic effect. Silver sulfadiazine has therapeutic effect, and all the silver dressings actually have the therapeutic effects on radiation dermatitis. So they consist of some topical corticosteroids. Using topical corticosteroids has some advantages, like they have anti-inflammatory actions in some conditions, and because of this property the wound can heal in a small time frame. But there are some disadvantages also, like it can cause skin eruption, and its efficacy is high when used twice daily. Means we have to use it for more times if we want to heal the wound properly within a particular time frame. And this can worsen the inflammation also. Using for prolonged time, it causes the contact dermatitis. But and also an uncontrolled use, it leads to its addiction. Means wound does not get healed within a proper time frame. Either you don't use it. It can be absorbed through skin and show some systemic side effects, like hypothalamic pituitary adrenal suppression, glaucoma, hyperglycemia, or hypertension. It can it can alter the constriction of blood vessels of skin also. So for wound to get properly healed, necessary uh, it is necessary that wound gets water, because a moist environment allows proper healing. Why? Because due to this, epithelial cells can migrate over the living tissues, and and the process can be accelerated. Sorry. And why it is important? It increases the wound healing up to 50%. There would be less scarring and better healing. There is enhanced and foster reepithelialization. It increases the cellular proliferation. It prolongs the presence of growth factors and cytokines. It promotes the angiogenesis, and there is a elevated collagen synthesis also, and with the lower rates of infection and wound cleansing and irrigation. So in this study, we have prepared the hydrogel for this to show the prophylactic effect on the wounds. So what is the advantages of hydrogel? It can absorb large amount of water of the wound, and it can also provide the water if it is required. It can swell and shrink, which controls the drug release. It can act as artificial extracellular matrix for cytokines, cells, the anti-inflammatory cells, the macrophages, and its porous nature is helpful in drug loading and drug delivery, and also the transport of gases like CO2 and oxygen. It helps in sustained release of drug for prolonged period of time. So then there is no need to use the hydrogel for 2 times or 3 times per day. It allows the transport of gases across the hydrogel to wound bed. These are the objectives of my study. In vitro study to elucidate the anti-radical scavenging potential of EP against gamma radiation induced protein damage and DNA damage, and a prophylactic effect of prepared hydrogel formation in radiation burn model in Sprague-Dawley rats. So the protein damage, we performed the protein damage and DNA damage study because it will give the better idea of radio protection, whether we should proceed to animal studies or not. So for the protein study, we used BSA as a model protein and calf thymus DNA and plasmid DNA as a model DNA models. In protein damage, we found that the SDS profile of BSA showed that at higher radiation doses of gamma radiation, it produces a very light intensity of the particular BSA band. But with increasing the concentration of our compound, the intensity of the band increased with increasing the concentration. And the same is shown in with the 500 gray radiation dose. But in case of the drugs increasing dose, there is no change in the intensity of the band. So it shows that the EP can show protection against gamma radiation induced protein damage. We also performed the spectroscopy studies with the radiation plus our drug doses. So it can be seen that the radiation, the treatment with our particular compound at different radiation doses, it retains the normal normal absorbance of the BSA. And also the fluorescence spectroscopy shows that beta-tyrosine level of the protein gets retained after treatment with our high concentration of EP, which is also showing similar results with at the 1000 gray radiation dose. Same is shown with the tryptophan concentration in the BSA. Like absorbance of tryptophan is the fluorescence intensity of tryptophan is retained after treatment with the compound after treatment with compound treatment. Same is shown in the tryptophan 1000 radiation at 1000 gray plus treatment with R EP. Similar results are shown in CD spectroscopy and BSA melting. It retained the normal melting temperature of BSA and also the normal absorbance graph of BSA in the presence of compound. These biochemical assays are also showing that after radiation and consequent treatment with our particular concentration of compound, it retained the normal concentration of carbonyl, the concentration of thiol in the BSA after treatment, while carbonyl content gets decreased because carbonylation is an indicator of damage. So it gets decreased after continuous treatment with the increasing dose of EP. So this paper has been got accepted in the International Journal of Biological Macromolecules. It has actually impact factor of now 8.2. Now we can proceed to DNA binding and DNA damage. Means the protection of EP conferred on gamma radiation induced DNA damage. First, we have performed sorry. First, we have performed the DNA binding because it shows that if the our compound is binding to DNA, then it can show some protection to it. So after performing the test or spectroscopy, we we can see that there is an continuous change in the intensity of the ellipticity of DNA after increasing concentration of DNA with ctDNA. It shows that the DNA is EP is binding to DNA and conferring protection. Same, the melting temperature of DNA is decreased after adding EP. It is showing that it is stabilizing DNA after unhelixing, unwinding of the helix of DNA, showing binding. Also, UV visible spectroscopy and the TM melting curve of ctDNA also showing the same result as the CD. Molecular docking is also showing that there is a small binding of EP at with minus 1.414 kilocalorie per mole at minor groove and intercalation with minus 2.117 kilocalorie per mole. Means EP can bind more by intercalation than to minor groove. These are some antioxidants performed using EP. It is showing that the MDA content was decreased in a dose dependent manner after giving radiation at 500 and 1000 gray, showing protection to the deoxyribose. And DPPH also showing that the normal inhibition of DPPH is increased at high concentration of EP. FRAP assay also showing the same results, that the concentration of ferrous ions increased in the different concentration of EP, showing that there is an anti-radical potential which does not causing the ferrous ions to convert to ferric ions. The plasmid relaxation assay also showing, first we elucidate the LD50 of radiation, which is 50 gray, which comes out to be 50 gray, and then we performed at this particular radiation dose in the presence of our compound on the plasmid DNA if there is more supercoiled or linear or open circular forms. With in a dose dependent manner, the CCC form, means supercoiled form, is retained at the high concentration, while linear form is depleted, while OC form shows some percentage, but that is not show some value. It is the graphical representation of the same results. Now I would like to proceed to the prophylactic effect of REP hydrogel. Because why we choose 2% EP hydrogel? Because while doing the therapeutic efficacy, we found out that the 2% hydrogel with 2% concentration of EP is more effective in treating the radiation wounds. So we proceeded with the 2% EP hydrogel for prophylactic activity. It is shown for this we have take standard and 2% EP and give hydrogel treatment 3 days plus 30 minutes prior to the radiation exposure. On day 1, there is no change in the skin structure, while at day 7, standard show erythema, while in 2% EP, there is no change in the skin. At day 14, it shows some erythematic appearance, but it gets healed within 7 days on 21st day. While in standard 21st day, the wound does not get healed properly, showing that there is some healing property using 2% EP because of its anti-inflammatory and antioxidant activities. So this is a graphical representation of the wound contraction and skin damage score, showing there is nil skin damage and the wound area is totally contracted after 2% EP treatment in comparison to the standard. And these biochemical assays also showing the same similar results, like in protein content it gets increased with 2% EP treatment. The hydroxyproline and collagen content also increased with this. The antioxidant assays also showing the same pattern, like MDA content gets decreased after MDA is an MDA is a radical, like which I have shown you in the DNA damage method. It is showing that the MDA content is decreased after treatment with 2% EP, and SOD activity and catalyst content increased with the treatment increased in the treatment of 2% EP. Histopathological analysis is also showing that there is an infiltration of inflammatory cells in epithelial and dermal tissues, while here epithelial tissue is also break, and which I think which does not get healed within 10 to 20 more days because of this inflammatory cells. And 2% EP hydrogel showing a limited number of inflammatory cells, and this epithelial cell lining is also healed. And this is a graphical representation of skin thickness. Here it is the size of epidermis, and this is the size of dermis, using the these particulars using measuring the thickness of these cells from this. Epithelial lining was measured from this to this, and in this from this to this. And dermis is this. Here is this. And there is also shown the arrangement of collagen fibers is good in both the tissues, while epithelial cells are here disrupted or erupted from the surface of the dermis. And there is an standard is taken as the 100% inflammation, because our 2% EP was compared according to standard in this study. And when compared both, there is a 10 to 15% of cells were there in the treatment with 2% EP. So to conclude, EP can protect protein and DNA from gamma radiation induced damage by binding to them, which confers the protection. And the 2% EP hydrogel was found to have a prophylactic effect against the radiation burn, which decreases the oxidants in the cells of the skin tissues. These are my some of the achievements here. And this is our research group. Is our director, and this is the division of CVR and defense, which helped me to perform these experiments or to complete my study. Thank you.
Okay, thank you so much, ma'am.
ma'am.
Thank you. Thank you.
Yeah, this was very much informative talk delivered by you from a farther apart from across the world. You
Thank you.
are
Thank you, sir.
वही कहते हैं ना, 7 समुद्र पार से ही आप बोल रहे हो, बाकी हम तो अंदर से ही हैं. Okay, and it is quite more early time. It is not easy for any scholar and student to just wake up at early morning.
Yeah,
I
it's
also
it's
don't wake
also
up at
minus
early
3 degree.
morning even. Yeah, with quite cold temperature, even though here about to 8 to 9 degrees Celsius temperature, but we are feeling very chilly. If you at this temperature, then I can assess how much cold was there. And is there also, you can say.
Yeah.
And
Yes.
now the topic is open for discussion. If any young scholar have any query, to raise your query so that madam will feel honored to reply over that. So over to young scholars for open house discussion. Yeah.
Hello.
Okay, ma'am. So nice to you for your nice presentation. Please accept this token of memory from our side, from the side of International Association of Zoologists, for your
Thank you.
kind
Thank you,
and talk. We will be in touch in future as well for your valuable
sir.
suggestions and experiences that will boost us in research and academia as well. So
Thank
we
you.
are very much and highly appreciative to you. You have accepted our invitation and given your valuable time and share your experiences with us. Thank you so much, ma'am. And
Thank you. Thank you, sir.
Jai Hind.
Thank you, Siddharth.
Over to Siddharth, please. Mr. Siddharth, please share the certificate once again. I didn't capture it.
Okay, sir. Please
Second,
wait.
you have. Yeah. For a while, you have to share.
Sir, just a minute, sir. Just a minute.
Yeah, yeah. Yeah, thank you so much, Siddharth. You can proceed ahead. Thank you so much.
Thank you so much, ma'am, and thank you so much, sir. Now our program, as per schedule, Miss Bhavna Paliwal.
Am I audible?
Yes, very much audible.
Okay. So the next presentation we have is of our Chief Organizing Secretary, Dr. Siddharth Da, currently joined as teaching assistantship. Hello, am I audible?
Yes, audible, very much audible.
Okay. So the next presentation is of our Chief Organizing Secretary, Engineer Siddharth Da, currently joined teaching assistantship cum PhD scholar in Dr. B.R. Ambedkar National Institute of Technology, Jalandhar, Punjab. He has recently worked on tissue engineering and biomaterial sciences. He received undergraduate degree in biotechnology from MMDU Maulana and postgraduation degree also in biotech from IK Gujarat Punjab Technical University, Jalandhar. He was also worked on acute dermal toxicity and decontamination study of carbon composite in Sprague Dawley rat models since 2021. He has more than 3 years of research experience in vivo study in nanobiotechnology, radiation, excessive wound healing, dermal toxicity study, and radiation decontamination study. He was working as research intern in Institute of Nuclear Medicines and Allied Science, DRDO, Delhi, National Cancer Institute, Kolkata, National Institute of Biologicals, Noida, as well as ICTC Lab MGM Medical College, Jamshedpur. He received PhD fellowship given by MHRD 2022, Best Young Scientist Award in year 2021, Shaurya Bharat Award in 2021. Engineer Siddharth Da has also honored by various science societies as MIAZ in earlier years and currently life member of IAZ, Vijnana Bharati, International Association of Engineers and All India Council of Technical Skill Development. He was reviewer of biointerference research in applied chemistry and reviewer management system, board member of International Journal of Creative Research Thought Grants. He has excellence in work in research activities and published various national and international research papers, review articles, and book chapters, including 30 plus review articles in reputed journals, which are available online too. Now, over to you, sir. All the best.
Thank you so much, Bhavna ma'am. I hope my screen is visible.
Yes, it's visible, sir.
Thank you so much. Now I would like to explain one very hot and very different area in biotechnology, that is bone tissue engineering and radiation prevention and decontamination in space engineering, as in astronauts. What is the major work in astronauts or space in bone tissue engineering or biotechnology. I am just talking about what is biotechnology for students of UG and PG, which is just joining recently. Biotechnology have actually many areas like food and nutrition, marine and aquaculture, industry, agriculture, environment, technology, medical, health, and diagnostics. It's also in biotechnology, they have two type of researches are available. That is primary research and secondary research. Primary research are three types. First one is the basic research like cell culture or normal lab works like biochemistry, genetics, or animal study. And next one is clinical research, that is phase 1 trial, phase 2 trial, phase 3 trial, and phase 4 trial. Mainly four types of clinical trials are there. And third one is epidemiological research, that is experimental or observational research. And second type is secondary research, that is only for meta-analysis or make a review paper. Now, my biotechnology broad area is medical and health diagnosis. And in that field, I am worked only in tissue engineering and biomaterials. That is from only waste product materials. And I want to this type of things are worked linked with space biomaterials, like these are also three types: habitation, astrobiology, and biomedical. In the field of habitation, that is only checked habit, suits, and coating. This type of worked in ISRO, India. But the other two types of work, astrobio and biomedical, this is like contamination types work and wound dressing, pharmacological work. These types of work are by only NASA or European Space Station or Russia Space Station. Only these three are worked on biomedical, like wound healing. And I only worked in tissue engineering. The main area is osteology, that is make a scaffold and the prevention of decontamination from radiation, that is only gamma ray. And I am worked done already in this type of work was done in DRDO in Mars. First thing ki why introduction, radiation exposure and contamination scenario. Why we can work on radiation exposure? Actually, we can see our conference theme is physiochemical, biological, radiological, and nuclear agents in human health. So first one is accident nuclear event. Like you can see in the last 50 years and hundreds years ago, that is Nagasaki and Hiroshima, we can see the nuclear events. And we can also see the one more in Delhi, Mayapuri, cobalt exposure. And next one is occupational exposure, that is like in normal industries waste. That's why this is the very important scenario for contamination by biomaterial waste product. Some types of biomaterials we can discuss, that is based on only carbon composite, that is graphene oxide, graphene oxide with silver nanoparticle, reduced graphene oxide with silver nanoparticle, and reduced graphene oxide with zinc oxide, and many more. These biomaterials also worked as antimicrobial property and as making a scaffold, that is in teeth implants, biomarkers and oral cancers, hydrophobic property. These are also work in a bone regenerations and increase the activity of antimicrobial property. First thing we can check ki all biomaterials mainly worked in for tissue engineering and radiation decontamination. The first property is very important. This is the antimicrobial property of the antimicrobial activity. So we can check ki these types of waste product biomaterials are so antimicrobial property or not. If they are not so antimicrobial property, then this type of products are not used in as a scaffold or bone regeneration techniques. Uh, we can also check radiation wound model. We can this is the Wavatron 2 by exposure of gamma 2 radiation, and we can make a silver nanoparticle medicine. You can see the last lecture by Dr. Deepti Sharma ma'am. She also worked in this medicine, antimicrobial and radiation wound healing by AZNP. This is the you can check the 7 group of the rats control. F1, F2, F3, and F4 are the different doses of silver nanoparticle, that is 0.2%, 0.5%, 1%, and 2%. Other one is standard. We can use the already purchased from market and vehicle. This is no medicine will take this group, and we can check ki 0.2% and 2% in silver nanoparticle groups are the soda waste result, and this some are the histopathological results. We can see ki the epidermis do not rupture, and the epidermis is already maintained after the treatment of 2%, but we can see the control, and in vehicle the epidermis and dermis are rupture. We can also study the toxic effect of antimicrobial of carbon composites. Why we can check toxic effect of any material? Because when we can decontaminate the surface of the skin or the surface of the any material. This is very important key. These material are non-toxic, because if the material is toxic, then the double infection are caused, because first thing key radiation cause also infection, and if the biomaterial who decontamination the radiation, these also affect the toxic. So we can secure the thing ki biomaterials are non-toxic, then it's used in decontamination efficacy. After we can check 14 and 28 days by OECD guideline, that is Organization for Economic Cooperation and Development guideline. This is two guideline by follow. One is accurate and second one is subaccurate. First, for accurate, we can check after 14 days, and for subaccurate, we can check after 28 days. And we can find that the carbon composites with a specific nanometer, we can use. Then we can find ki no rats are mortality, normal pulse rate and breathing, normal food consumption, normal physiological behavior, normal body weight, normal urine, and no salivation we can found. After 14 and 20 days, 28 days, we can dissect the HD rat, Sprague Dolly rat, and we can check the histopathological results and biochemical results of the HD rats, and we can found ki one more thing, we can also check 14 and 28 days ki dermal irritation study and sensitization study, that is guideline followed by 106 and 110, and we can found as the drug does not show any irritation and any sensitization scores or any rate or any grades. This is the some biochemical parameters we can found after 14 and 28 days. The result shows the all group of the animal and all the carbon composites shows normal biochemical parameters, and it also shows that key that biomaterials is non-toxic. This is the some histopathological parameters from the HD rats of kidney, liver, spleen, and skins. We can found that ki to 500 mg per kg body weights rat and 200 mg per kg body weights rats, the skin of the layer of the skin, dermis and epidermis are found constant, but in the group of the control we can found the rupture of the dermis and epidermis layer, as well as we can check 2000 mg per kg body weight. We can just see that it's make a tumor-like structure in the histopathological results. And in the kidney cell, we can say the BC are the just common Bowman capsule cells are constant, but in control group, the Bowman capsule cells are also rupture. In also see in lungs, alveolar cell also constant in 200 mg per kg body weight and 500 mg body weight, but in 2000 mg per kg body weight and control group, we can see the alveolar cell are also rupture. When we can found that the biomaterials are non-toxic, then we can check the efficacy of the decontamination of radiation by the apparatus of France diffusion apparatus. In this apparatus, we can just stick the sample, stick the skin of the sample, and after 5 minutes we can just apply the radiation operation, that is technetium, and 45 minutes we can check by the atomic absorbance spectroscopy, and the result is very shocking because the penetration of the biomaterial is just below 0.3, that is 0.3 ppm. That is very low penetration of the biomaterial, and we can show that this is the very good biomaterial for decontamination. After that, we can check the decontamination efficacy by absorbing of the cotton. The cotton is dissolved in the carbon composite emulsion, and we can just wipe the surface of the skin on the rats, and we can found the control group that is only 10% absorbance, that is the cotton is dipped on water and just wipe the radiation, and the cotton absorb only 10%, 10 to 15%. But when the cotton is dipped in the carbon composites, that is 1%, 2% and 3%, and then we wipe the surface of the skin, then we can found that 75 to 80% radiation will absorbed by the cotton, and normal cotton just wipe the, then it is so 50 to 55% of radiation absorbed. After the result, we can conclude the material is non-toxic and base decontamination efficacy. The penetration is less than 3.5 microns, and we can found no harmful effect of carbon composite. that is specific nanometer, we can say already. And Dr. Mahesh Kumar Sa and Dr. Adesh Kumar Saini sir helped me in my PhD work, and we have already continued this work in my PhD. But this time we can derive the carbon composite by plant waste product, and we can also aim to use this product are used in sunburn or wound healing efficacy, like fracture bone wound healing models we can use. But my aim is future ki after searching the some website and NASA and site of the ISRO, we can found that ki this type of biomaterials are or bone tissue engineering are used in very much in space stations. We can see these two type of space suits. First one is female astronauts, and second one is suits for male astronauts. And after putting this dress, we can find many types of medical problems, like women suffer hearing issue and advancing age and do not display the waist two sides hearing in the left ear. We can also see the clinical significance for visualization impairment. Female astronauts also susceptible osteostasis intolerance. We can found a problem in urinary tract infection for male and female also. Kidney stones are commonly found in females. In male, we can found the immune system are weak in the bones. But actually, this type of problems we can see, and also I am medical conditions. We can find 100 types of medical problems are seen in astronauts who live in 0 g, means zero microgravity in space station. What is microgravity? In Earth, the microgravity we can say 9.8 meter per second, and this is so key 1 g, and 1 g means 9.8 meter per second, and at in the moon surface we can find 0.18 g, means only 0.72, sorry, 0.88 less than Earth's surface. And in the surface of Mars, that is Mars planet, 0.38 g. This is also very less. And International Space Station, the G is 0. And this is the problem of microgravity. We can found in the major problem. We can see some visualization effect. We can see the next slide due to microgravity. I hope slides is proper visible. My slide is proper visible. Hello. Hello. Okay. Thank you. Some visualization effect due to microgravity. When in the surface of Earth, the force look outside, and in the when we can see zero gravity, the force are repulsion each other, yani oxygen repulsion each other. And we can see the second figure. On the surface of Earth, the blood or the fluids inside the body flow continuously in upside and downside, that is in 1 g, the normal fluid movement, no movement is changes in the surface of Earth. But when we look on the zero gravity, we can see the visualization effect of the downside the figure. The fluid is only through move on side the brain. The leg is 0% fluid, and very minimum fluids are go inside the leg. And we can also see the last figure. This is the brain. The fluid towards the head cause increase internal pressures. And we can see that this figure also for cornea, fluid pressure cause optic nerve compression, flattening on the back of the eye wall, and optic disc swelling. This is the main effect of microgravity. Some other effects we can see in space, that is exposure to high level of radiation increase the risk of DNA mutation, degenerative disease, construct and lifetime risks of cancer. In case of bone, insufficient bone formation compared with bone restoration reduce the bone mass and bone strength, leading to an increase in fracture and osteoporosis risks. Due to immunological disorder, the immune system is weak in the ISS, International Space Station, or in zero gravity. Lack of gravitational loading and reduced muscular activity result in muscular atrophy accompanied by functional and structural alteration. We can see the main problem also caused by body fluids, that is headward shift of fluids. All fluids, maximum fluids are shifted towards the head, including blood, leads to compensation, cardiovascular system change, decreased leg volume. Leg volume is decreased due to the all fluids are shift towards the head. That's why the volume of the leg is decreased, and even long-term ocular damage causing vision problem. Uh, this is the space suit. Space suit is made by 9 to 10 layers. That is HT1 outer layer, and second layer is the micrometeoroid protection. Third layer is thermal layers. Fourth layer is micrometeoroid protection, registration net, pressure bladder, comfort layer, undergarments. And we can see that this suit is made by biomaterials. Then I hope the this type of biomedical issue are resolved by biomaterial making suits. This is the conclusion and future work: biomaterial derived radiation protection astronaut suits, microgravity and impulsion for wound healing, scaffold based tissue engineering in microgravity, space radiation decontamination soap and detergent kit. And this type of work I should work in future. This is my few publications. I am acknowledged our guides and supervisors. This work was conducted in DRDO INMAS, so I acknowledge our research team. This whole our research team in INMAS DRDO. And thank you. This is the session for question answering. If anyone want to ask any question related to this PPT, then you should ask. Anyone asked to any questions? Actually, LD50 dose we can calculate by body weight. Trita Pratima Saha, you asked any question?
No, sir.
Okay, okay, thank you. Dr. Sushil Kumar Upadhyay, sir.
Yes, sir. Yes, sir.
Sir, may I proceed? The presentation slot start now.
Yeah, you can. If you wish to make a break of 5 minutes, then you can. Otherwise, you can proceed ahead.
Okay, sir. Okay.
You have to ask the organizers if everyone is ready and no need of the break, then there's no problem. Otherwise, no need to break the meeting. Just hold on for 5 minutes. Take
Okay,
a
sir.
break.
Okay, okay.
And
Sure,
then
sir.
you can proceed. Okay?
Okay, sir.
Yeah.